Okay, happy Monday. So we have an exam coming up a week from today. So I kind of looked at the homework and see how you're all doing. There's a number of you, it's time to start that homework now, so it's not so painful later. So that exam is going to cover chapter 12. Remember, that was our eukaryotes, our fungi, protozoa, algae, a little bit. Chapter 6, growth. We have ways of describing growth and ways of counting bacteria, things that they need to grow. 5 is metabolism. And then I'm thinking that we will get pretty close to finishing up chapter 7, genetics today. We may need to bleed into Wednesday's lecture to finish that up a little bit. And then we'll start viruses, but the viruses will not be on Monday's exam. So I think your homework assignments, I guess, pretty well there for when they're all due. So remember, homework's due Monday, time of exam. I have to cancel that hour that I usually have office hours Wednesday morning, dentist appointment. I still have Tuesday and Thursday afternoon office hours, 12 to 2:15, and I'm going to be here Friday. So if that works for anybody, if you still haven't come and look at your old exam, you're welcome to come still do that. Or if you got questions or want to go over anything, then please do. Okay, so we have been following the flow of information. So we've talked about how replication happens, and then how RNA is made from DNA, transcription. You don't have to have replication necessarily before you have transcription. I've run into that a few times. Students thought that you always had to have replication first and then transcription. You have replication when the cell copies its DNA when it's getting ready to divide. But you would have to have maybe lots of proteins made from that DNA. So you need, lots of transcription and translation from that same strand of DNA. So just keep that in mind. We talked about in transcription, we have initiation, elongation, and termination. And now we're going to talk about kind of those same initiation, elongation, and termination of translation. So it's easy to get those two things mixed up. Before we get into translation, there's a little we need to understand about the genetic code. Right, so this code was deciphered by Marshall Nirenberg, working at the NIH in 1961. And of course, what the genetic code describes is how we have codons of messenger RNA. That's what here, top and both sides. We have 3 bases of messenger RNA that make up a codon, and how which ones come together and which amino acids will result when we have, a certain sequence together. How did Nirenberg decipher this? A lot of trial and error. He did radioactive labeling of amino acids, and just again, trial and error, figured it out. So if you have 4 bases, and you put them in groups of 3, there's actually 64 different combinations that we could get. But of course, we only have 21 amino acids. So there is actually some wobble in this code, or also what we would say degeneracy, right? If you call somebody a degenerate, that's not a very nice thing to say about somebody. But when we're speaking technically, if something's degenerate, that just means that it's lacking distinctiveness in structure. So essentially that means the 3rd base of the codon, sometimes it doesn't matter what that 3rd base is. So for instance, for serine, no matter what that 3rd base is, if it's U and C, then that's that codon will encode for serine. Okay, so sometimes that third base matters, sometimes it doesn't. Obviously, don't memorize this. You should be kind of aware there is one start codon, and that's AUG, that maybe one thing that's reasonable to kind of understand, that encodes for formal methionine in bacteria. And then there's 3 different stop codons that when those codons are read at the ribosome, there's not any amino acid that will match up with that. So that's what causes the termination of translation. Sorry. So we'll look at that a little bit more closely. Okay. All right, so steps of translation. We talked about before the different kinds of RNA. We talk a lot about messenger RNA. Of course, ribosomal RNA is what makes up the ribosomes. There's also those tRNAs, and the tRNAs, their job, we talked about that before, is to carry amino acids, the correct amino acid to match up with that messenger RNA sequence. When the tRNA is carrying an amino acid, we consider it charged. All right, so we're actually going to skip over the process of how that happens. It's mediated by kind of a group of enzymes. Your book doesn't go into that, and we're going to skip over it. But just when you're talking about a tRNA that is loaded with that amino acid, then we say that it's charged. Okay, so we're going to skip over that step and jump into initiation, elongation, termination. And then there's just a couple comments that protein that gets made isn't necessarily functional right away. So just a few comments there. All right, so that's steps of translation. We'll start with the figures that your book gives you. And I want to add in one of my own that I'm hoping will clarify a little bit. And by the way, there's a really good video tutorial that's part of your homework. It's one of those Dr. Bauman's video tutorials. It's like 4 or 5 minutes long. It's just a really great kind of rehash of how this works. So don't watch that like triple speed. Go through that again. That will be helpful. Okay, so translation. This is taking place at the ribosomes, right? But actually, remember, the ribosome is composed of both the small subunit and the large subunit. So it starts out with the small ribosomal subunit. And remember, your book uses these little blue icons. That's the tRNA. All right, it's already charged. It's got a formal methionine on there carrying it. And remember, the anticodon is part of the tRNA, and that's the complementary base pairs to the start codon here on the messenger RNA. All right, we have different sites on the small ribosome. Okay, so the P site, that stands for the peptidyl, or we'll say the peptide site. That's the site that holds the growing peptide. And it's also where the first amino acid docks. All the other incoming amino acids are actually going to dock at this A site on the small ribosome. And that A, we'll just say that stands for acceptor. Okay, so we'll say the rest of the new tRNAs dock here. So we've got the first amino acid, formal methionine, coming in there. Takes a little bit of energy here in the form of GTP to catalyze that kind of this reaction here. So that comes into, again, the P site. And once that binds to the P site, then the large subunit of the ribosome will come together and form what we call the initiation complex. Okay, now translation is ready to begin. I like this other figure that I got from another textbook, and actually added a little bit onto it myself, because this helps us understand in a little bit more detail how important it is that that messenger RNA docks at kind of the just right spot for the ribosome. Okay, so this figure is a little bit, usually we draw the small subunit on the bottom. For here, whatever reason, they drew it up on the top here. And remember that small subunit of the ribosome, remember what it's made of. It's made of strands of ribosomal RNA along with some proteins. So here's a strand of that ribosomal RNA. And again, this is the small subunit right here. And it's important that the messenger RNA docks at just the right spot, because this formal methionine needs to dock exactly into that P site. Okay, whenever we have a sequence of base pairs that's really important, we like to give another name for it. Remember, we had the Pribnow box in transcription. That was that -10 region where it's really important that the messenger RNA, the RNA polymerase binds. Here we have this segment called the Shine-Dalgarno element. Again, and this is on the messenger RNA. In your book, we uses that term maybe once or twice. Okay, this is bacteria only. Okay, eukaryotes don't really have the same kind of sequence there, even archaea. And that binds to certain sequences on that 16S ribosomal RNA, so that the AUG, that start codon, falls exactly on the P site. You can see if this didn't match up quite right, then, and if this was, off by 1 and you had UGC instead, okay, the right amino acid's not going to come in there, right? I don't even know if translation would even begin, or it would just be making a totally different protein if it was off a reading frame. So we got to get that. Oh, there we go. Okay. So we have to get that docked just right. Remember, we talked about after transcription for eukaryotes, they have some modifications that happen. They have a 5' guanine cap. Okay, so for eukaryotes, it's that cap that kind of lines up just right. So Shine-Dalgarno element, important in initiation of translation. The directionality, that was the other thing I wanted to point out. So this is going to start at the 5' end of that messenger RNA. And that's easy to remember. I drew this part out here myself, so it's a little bit sketchy there. But that's easy to remember because actually this messenger RNA may not be done getting transcribed. Okay, it can still be, here's the RNA polymerase here that's working on the DNA. This is the 3' end. Remember, it's always adding on those ribonucleotides onto that 3' end, the 3' hydroxyl group. So that could still be getting transcribed. And so it's the 5' end that's fitting here at the ribosomes. Okay, questions there? Don't be afraid to stop me. All right, so that's our initiation complex. And then getting back to your book's figures, elongation, basically you have the charged tRNAs that coming in, they're going to come into that acceptor site. And then there, it will, very briefly, the growing peptide will kind of switch over to that new tRNA. And that peptide bond formation catalyzed by ribozyme. Remember what ribozyme is? That's ribosomal RNA that acts as an enzyme. We said ribozyme can catalyze lots of reactions in that translation process. Okay, so just for a moment, that peptide is transferred to the A site, but then very quickly, this is got a kind of like a ticker tape that's pulling through the ribosome. It switches into the P site. Okay, so We usually describe that growing peptide as resting at that P site. And then this just continues on and on. So here's a new amino acid. Again, the anti-codon on the tRNA matching up with the codon on the messenger RNA. Get peptide bond formation. And then when the tRNA has gotten rid of its amino acid, it moves into the E site, also known as the exit site there. And then it can go and get charged again, get another amino acid. Okay, let's look at this animation. Let's see if I can get it to work. Oh, there it goes, just slow. Okay, it's not coming up for me. It was going to, I think maybe our internet's a little slow in here or something. So doesn't want to do that. If you have a chance, take a peek at that animation. It's kind of a, somebody's made a cartoon of the translation process. And if you start at about 2 minutes, it actually, the beginning of it is transcription. And then at about 2 minutes, it has the messenger RNA leaving the nucleus of the cell. So we're talking about prokaryotic translation. It has a lot of similarities to eukaryotic translation, but it has the transcript leaving the cell, and then it gives you an appreciation of how quickly this all happens. We think that the speed, about 15 amino acids are added per second. All right, so it doesn't go quite that fast in the animation, but it kind of helps you appreciate how fast this all happens, and it's pretty amazing, actually. Okay. All right, so again, if you have a chance, look at that. Oh, maybe I'm going to show you just a little bit of it here. Okay. Get rid of that. Oh,
the innermost work.
We gotta get rid of that over there. I'll just show you just a little piece of it here.
Part of the cell.
Oh, there we go. There we go.
Then,
So
in a
there's the
dazzling
transcript
display of
leaving the
choreography,
cell. Yeah.
all the
Pretend this is
components of another
prokaryotic
molecular
cells, but
machine
they're not eukaryotic.
locked together
That
around
big
the
blue
RNA
blob
to
is
form
the
a
small
miniature
ribosome.
factory called a ribosome.
The big,
It translates the
subunit,
genetic
light
information
subunit
in the
so this
RNA
is the
into a
messenger
string of
RNA.
amino acids that will
Secret
become
and all
a
those little
protein.
things flying around the tRNAs.
Special
Large
transfer
tRNA.
molecules,
So the
the
red
green
part is the amino
triangles,
acid.
bring each amino acid to the ribosome.
Full amino acid on that.
The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine.
And in a second, you'll see kind of a red.
Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape.
That little red ribbon, that's the growing
code for
polypeptide
each amino
chain.
acid is read off, 3 letters at a
So
time,
it just kind
and
of
matched
get an idea
to 3
of what's
corresponding
going
letters on the
there.
transfer molecule.
Yep. All right, and also that you can have that one messenger RNA. So the red line is the messenger RNA. You can have that same messenger RNA being translated by lots of ribosomes all at once. Okay, we call that a polyribosomal complex. Just kind of, I think that happens more often in prokaryotes, but that can happen in eukaryotes as well. How does that growing polypeptide get terminated? How does translation stop? Your book doesn't give you a picture to go along with that, so it does describe it in the book, but I always like to have a graphic. Basically, remember in transcription, we had either the rho protein, that was involved, or it was just written into the sequence of DNA, where you had the palindromes and the hairpin loop that dissociated the messenger RNA or the RNA from the DNA. Here, this involves a stop codon. So here's that UAA. Again, you don't have to memorize what those stop codons are, but that when that gets to the ribosomal complex, there's a pause because there's no tRNA that matches that. Okay, so the stop codon is read. Again, no transfer RNAs will recognize. So proteins called release factors are going to come in and bind right here to that stop codon. And that's going to activate ribozymes. To sever that polypeptide. From the ribosome. And then you can see in the third picture here, the ribosome dissociates. Okay, the small subunit breaks apart from the large subunits. tRNAs go their way. So that doesn't necessarily mean that now this protein that was made is necessarily functional. Okay, it might not be in the best interest of the cell for that to happen because it may be, you may have something like a toxin that's being made and you don't want that functional right away. So in some cases, certain changes need to be made to actually activate that protein. If it's an enzyme, maybe there are cofactors that need to be attached. Remember, cofactors are some inorganic ion, maybe something like copper that needs to bind to the enzyme to make it able to function, to catalyze reactions. Remember, if that cofactor is organic, what do we call it? And it's a coenzyme, right? Typically, that very first amino acid, that formylmethionine, typically is cleaved off. And there's a number of reasons why that may happen. We know in the case of for bacteria that are trying to infect hosts, that formal group is a big tip off to the host, so the host immune system, that this is something that's foreign. So that might be one reason that formylmethionine is always cleaved off. There might need to be some further bonding that takes place to make a more complicated structure in order for that to activate, or it could be some kind of cut, for example, that needs to happen to activate it. So just some examples of what happens then once that polypeptide is made. All right, so I like this summary figure that an old edition of your book had. For some reason, the new edition, it's a little bit simplified, but just kind of keep in mind what's going on here, that if you look at the DNA, the template DNA strand, remember the template is you got two strands of DNA. The template one is the one that's used, the RNA polymerase that actually transcribes it. And it transcribes that promoter region. Remember, in prokaryotes, that gene can be polycistronic. It can code for several different genes. Not necessarily will, but it could. And then that gets transcribed into the messenger RNA. So you've got, the start codons and the stop codons. All that information is written right in there into the messenger RNA. It gets read at the ribosome there. And what you end up with is 3 different proteins gets translated at the ribosome. So again, kind of good, nice overview there. Okay, I meant to skip that, so never mind that. Okay, Let's do a practice question. This is kind of wordy. Some of my exam questions can be wordy. I try not to make too many of those. So I'll give you a chance to read that. Remember I said on the exam, it's very easy for me to try to confuse you the difference between replication and transcription and translation. So I'll give you a second to read that. Put that down somewhere, which one you choose, or tell your neighbor which one do you think it is. All right, so I was so nice as to bold these words. They might not necessarily be bolded on your exam. So you're looking for the one that's true about transcription. All right. This is not true, only one strand or the other. And remember, we had names for that, whether it was, the plus side or the minus side or the template or the coding. So I want to look back at those if that's still murky in your mind. Transcription begins with the Stein-Dalgarno sequence. That would be correct if it said translation, but it doesn't. Remember, RNA polymerase, it's built into it a helicase function. So it unwinds the DNA strands itself. In replication, DNA helicase is necessary. This one would be true if it said replication. It begins at the origin. Okay, remember, transcription begins at the promoter region. Initiation begins at the promoter. So the correct one here is the sigma factors are crucial to initiate, to get RNA polymerase to bind tightly to that promoter region. Any questions about that? It's good. So another nice way to review, this is a good table in your book, just to kind of make sure you have the basics about replication, transcription, and translation. Cover that up and say, let's see, do I know what's used as a template? What do I call the start site? How often does it make mistakes? That's what fidelity mechanism is asking. So we talked about pretty much everything here except maybe this direction of translation. We didn't really worry about that. Everything else, that's good. That's good review. Cover that up. Be as active as you can. Don't just read over it. Test yourself and see if you know it. All right. So it's regulation. So this is related to what we've been talking about, because, well, why do we want to regulate? Making proteins is energetically very expensive. Okay, so if we take a, say, average protein that's 300 amino acids long, how many ATP do you think a cell has to spend to make an average protein? Somebody yell out a number. Wild guess. Pretty good. 1,000. Anybody have another guess? 30? That was it. Somebody last semester guessed 1 million. 1,000 is pretty close. The number that I have come up with is that this costs about 1,200 ATP. Okay, so that was a very good guess. So energetically, when you think about, we talked about during glycolysis, the cell gets 2 ATP. So it's gonna have to do a lot of glycolysis, a lot of fermentation, if that's all the cell does. If it respires aerobically, it's gonna get, or anaerobically, it's gonna get a lot more. But so that's a lot of ATP. We don't, the cell, the bacterial cell does not want to make proteins unless it has to. So what are some mechanisms that it can use to regulate and not make proteins if it doesn't have to? Well, there's different kinds of regulation. We could look at consensus sequence variation of that promoter region. So we did talk about that a little bit before. Just remember, and I'll come back to that. Remember this slide, and we talked about how the different operons of E. coli, if you look at their sequence, that -10 region Cribnell box and the -35 regions were really important. And depending on what those bases were, the RNA polymerase is going to bind more or less tightly. So the cell, kind of knows if this is going to encode for proteins that need to be used a lot, then those bases are going to be pretty close to some consensus sequence, and the RNA polymerase is going to bind almost all the time. Okay, if that sequence is not as close to this, then it will be transcribed less often. So there's a little bit of regulation just written into the DNA. A cell can use different sigma factors that will bind the RNA polymerase more or less tightly to that promoter region. But what we want to focus now is operons. Okay, and operons, we mentioned this before, this is when genes are regulated together. Okay. And it's really a very efficient way to regulate how much enzyme is made, because often the enzymes for a particular pathway will all get regulated together, which just makes sense. Okay. Again, this is really a prokaryotic thing. There's some gene clustering that goes on in eukaryotes, but it's not the same as what the bacteria do. Okay, so I know that you've talked about operons before. It might have been a while since Bio 120, or I don't know, maybe you talk about it in genetics. I don't know. But a lot of students don't like this. Don't try to memorize this. I'm going to give you some examples. We're going to work through some examples of different operons, and it's really going to help you to, this sounds corny, to think like a bacteria. Okay, so to think, okay, in what situation would these structural genes need to get translated? In what situation does a cell need those proteins, and in what situations does it not? So that's really going to be key. So, and don't forget what we've just talked about with transcription. But this is the DNA strand here, right? There's a promoter region. Remember, that's where the RNA polymerase is going to bind. There's a sequence of bases just downstream there called the operator region. This is where any repressors will bind. Okay, so think about it. If there's some kind of repressor bound there, is the RNA polymerase going to be able to move down the strand to get to those structural genes? No. Okay, so that's going to stop transcription if there's a repressor there. Where does the repressor come from? There's some kind of regulatory gene. It's somewhere else on the DNA where that repressor, that would encode for the repressor. Okay. All right. So let's look at some examples here. Operons are really going to fall under either what we would consider negative control or positive control. So I am kind of organizing the information here a little bit differently than what your book does. I'm going to talk about this, but I don't organize it quite the same. If an operon falls under negative control, that just means that it involves some repressor. Okay? So there's two kinds of negative control. There's an inducible operon, and there's a repressible operon. Okay, so this is the part that gets confusing without looking at specific examples. An inducible operon, this is usually going to be off, meaning that the genes are not transcribed. Okay, so in other words, there's something present that's going to, that would have to, usually the repressor is there on the operator and something would have to be present to deactivate that. Usually catabolic pathways are regulated this way. So remember, catabolic, that means that it's breaking down some nutrients, usually into some smaller metabolites. Okay, so our example here is the lactoperon of an inducible operon. So we're going to look at that in just a second. We're also going to look at a repressible operon, and a repressible operon is usually going to be on, meaning it usually gets transcribed unless there is a co-repressor that would activate the repressor to make it come on. Okay, so usually the repressor is off, usually it gets transcribed, and these are more likely anabolic pathways. So some biosynthetic pathway to make something like an amino acid. All right, so the example that we're going to use is the tripoperon, and that's going to control the enzymes that are necessary for the cell to synthesize the amino acid tryptophan. Okay, so again, keep in mind when the cell would want to, make these proteins and when it wouldn't. Okay, so I found this little diagram of the lactoperon and I thought it might be useful for us to label it together. Okay, hopefully you could do this yourself, but we'll just go ahead and do it together. All right, so this big blobby yellow thing, that is supposed to be, I'll write it over here, that's the RNA polymerase. And remember this number 3 that it's bound to, the place on the DNA where it's bound, that's the promoter region. Okay, what's in its way? That big green block, that is the repressor. And remember, the repressors bind to the operator region. So number 4 is the operator. Okay. So then the structural genes that, in this case, in the top of the picture here that are not getting transcribed, let's see, 6, 7, and 8. 6 is LAC-Z, which is encoding the enzyme beta-galactosidase. I'll just call it beta-gal. Okay, beta-galactosidase. That is the enzyme that cleaves lactose into its 2 monosaccharides, the lactose and glucose. Okay, so that's really important enzyme. Number 7 here, that's LAC-Y. That is a permease enzyme. So an enzyme that's a permease makes it more easy for a substance to enter into a cell. Okay, so in this case, the lac permease enables lactose to very easily get into a cell. Now, it can get in anyway, but the analogy I like is that lac permease is like the fast pass, right? So if you've driven around Chicago and you got a fast pass, you go right through the toll booths. You don't even need to slow down. Okay, if you don't have a fast pass, then you gotta stop and you gotta pay higher tolls even. So permease just lets the cell lactose enter the cell really easily. And 8 here, this encodes for lac A, which is, I have to look this up, thiogalactoside transacetylase. Okay, we are not really sure the function of that in lactose metabolism. We think it actually may have some kind of detoxification function. As far as the last I read about that, we don't have a clear picture of that, so we won't worry about that. Okay, but the idea is that these are enzymes necessary for the cell to metabolize lactose. So enzymes necessary for cell. Okay, and of course it would want to do that as an energy source. What's different about the bottom half of this picture? Those little number 5 puffballs, I'll just write that in up here. That is referring to lactose. Or sometimes we say allolactose. It's just a little different linkages. That's the only difference between lactose and allolactose. The point is that when lactose is present, then that binds to the repressor here and deactivates the repressor. So now the RNA polymerase transcribe those proteins, transcribe and then eventually they'll get transcribed into messenger RNA and translate them into the proteins necessary to break down lactose. Okay, so it's a great system. You don't want to make the enzyme like at the top unless what you're trying, the enzymes are for, is present. Now there is another layer on here that we'll get to in a minute. If glucose is present, then the cell really doesn't want to use lactose. It would rather use glucose. So there's a way to, even if lactose is present, for those genes not to be transcribed. So we'll get to that in just a second. Okay, the other example of negative control that I want to mention, and there is a little animation in your homework that goes through this, it's the trip operon. So the trip operon encodes for 5 different proteins that are necessary for the cell to synthesize its own amino acid tryptophan. Okay, so that's the product here. All right, so a cell is normally going to need to do this unless it already has an abundant supply of tryptophan, or if it just makes too much. Okay, if we grow E. coli in a medium that already has tryptophan in it, cell does not want to keep making its own tryptophan. It's just going to use what's available. Okay, so tryptophan actually acts here. It acts to activate the repressor. So we say tryptophan is a co-repressor. In this case, it will bind to the repressor and bind at that operator region to block transcription. Okay, so again, we don't want the cell to be making tryptophan if there's a bunch of it there already, so conserve energy. All right, so back to that lac operon, this is the best example that I could tell you of positive control. An operon that's under positive control means that there is some activator that the messenger RNA has to recognize in addition to there not being a repressor in the way. Okay, so an example is the CAP protein, catabolite activator protein, and the cyclic AMP complex. All right, so like I mentioned before, if there's glucose there, then the cell does not want to bother to make the enzymes to metabolize lactose. Okay, so it says here, the levels of cyclic AMP in the cell are related to the amount of glucose in the media. Okay, so if we have a lot of glucose in the media, then these two things, glucose and cyclic AMP, are actually inversely proportional. So if you have a lot of glucose, then in the cell, there's going to be the cyclic AMP levels are going to be low. Okay, and if there's low cyclic AMP, so there's not going to be any cyclic AMP bound to that catabolite activator protein. Okay. But if glucose levels are low, the levels of cyclic AMP are going to be high, and you will have then cyclic AMP, that cap complex. Okay, and then lac is going to, those lac genes are going to get transcribed. Okay. So yeah, so that's the activator for the lac operon. Okay, so put that together with your book's picture here. Here it's got cyclic AMP bound to cap. And this makes more sense when you understand the RNA polymerase, this is a big enzyme. It has to recognize that as well. And once that cyclic AMP cap is recognized, then transcription is going to proceed. Okay, and remember that's only going to happen when there's no glucose or very low glucose. Okay, and of course there's lactose present as well, because then that will get rid of the repressor. Okay, so if you want another review of that whole lac operon, this was the best kind of review of that I could find on YouTube. It's about 5 minutes long, and it talks about both the negative control as well as the positive control with the activator. Okay. One more thing I want to say about that, and this relates back to our growth curve that we talked about a few weeks ago. Really what we're describing here with both the negative control and the positive control, that's catabolite repression. Okay, so glucose and lactose, they're catabolites. They're things that the cell will catabolize. And with those two, negative and positive control, like I said, you can repress using one catabolite, lactose, in favor of using another, glucose. Okay, so if you say start E. coli in a culture that has both glucose and lactose, okay, then this is what your growth curve will look like. And this is taking optical density readings. Remember, that's with a spectrophotometer. We talked about that. So you're just putting your, say test tube in the spec and testing for the cloudiness. Okay, the more growth, the more cloudy it's going to be. So when you first test that, or when you first start your culture here, inoculate the E. coli, you're always going to have a little bit of a lag period where growth is slow. But once it starts growing exponentially, and when glucose is utilized, you're going to have 1 log period. Once that glucose runs out, you have a second lag period when the cell is gearing up to use lactose. Okay, and then you have a second log period when lactose is utilized, and until it runs out, or until there's too many waste products or whatever. Okay, so this kind of curve we describe as a dioxic curve. Dioxic means two-phase. All right, so one log period when it's using glucose, a pause, and then another log when it's using lactose. So it's kind of fun to see that. It takes a number of hours to actually see that in the lab. So we don't have time to do that in Bio 120 in a 2-hour class period, but you might do that in an upper level course. I'm not really sure. Okay. All right, questions, anything about operons? Okay, chapter 7 closes out just thinking a little bit about mutation. So in this process of, a cell, passing, during the flow of information, during replication, mutations can happen. Things can go wrong. A mutation is a permanent change in the DNA or RNA. Usually it's going to be bad for the cell. Okay, like overwhelmingly is going to, cause a cell to be less fit, but maybe every once in a while a mutation will happen because bacteria, replicate so frequently, one that will give the cell some kind of an advantage. So we're going to talk a little bit about what happens with some point mutations. So we're going to spend a little bit of time here. Your book barely mentions at the very end of the chapter some transpositions and inversions, just a little bit. We're not really going to get into that, but just be aware of, mutations can happen more than just, one base, basically. So we're going to look at some terminology associated with mutations and kind of understand why they're bad. We're going to look at some things that can cause mutations. What does the cell do about it? A few repair mechanisms, and then to finish off, another way that a cell's DNA can be altered, and that's through horizontal gene transfer. All right, so long chapter. We're approaching the end of it. This is the figure that I posted in your notes. And I'm gonna kind of break this up so it's not quite so little on my screen here. So the first part here, say we have up at the top here, this is, say, our normal DNA strand, or the one that's being transcribed. Here's the transcript and the resulting proteins that we would normally see. Okay, what if this base right here, maybe DNA polymerase 3 made a mistake and put the wrong base in there? Of course, that doesn't happen very often, but maybe it did. Maybe, remember, this should have been an A and it put in a G instead. So the resulting messenger RNA transcript would have a C. And then, but because there's wobble in that genetic code, actually encodes for the same amino acid, phenylalanine. Okay, so even though there was a mutation, we call it a silent mutation because it didn't make any difference in the actual amino acid that got translated. Makes sense, hopefully, the terminology there. Let's say this guanine right here was instead a cytosine was put in. And then we would have a guanine there in the mutated messenger RNA. So now instead of alanine, we would have the amino acid glycine. Okay, so that makes sense in terms of it corresponds to an amino acid, but it's not the right sense. Okay, so we call this a missense mutation. Okay, instead of alanine, we got glycine instead. Will this protein still be functional? Maybe, yes. It depends if that amino acid was at a crucial spot or not. Potentially not. It could, even one amino acid, make a difference into whether that protein is functional or not. Okay, so we don't really know. What if this adenine right here was actually changed to a thymine? Then instead of tyrosine, we would get UAA, which is a stop codon. Okay, so that means that the polypeptide will prematurely stop synthesis. Now, this is probably going to be very bad. You're probably not going to get a protein that's going to function. So we call this a nonsense mutation. Okay, so however you can, fix in your mind the difference between missense and nonsense. If this happens at the very end of the polypeptide, it may not make any difference. But if it happened at the beginning, obviously you could have, a couple hundred amino acids that never got translated. So it's probably going to make a pretty big difference as to whether you got a functional protein. If you had an insertion or a deletion, you can imagine that that would be pretty bad. If this thymine got inserted in there, that's going to throw off the reading frame for the whole rest of the polypeptide. So after that, you're going to have likely all different amino acids. So this is going to be a major change in the amino acid sequence. And so again, a frame shift, any kind of frame shift mutation is probably going to be pretty bad. So same thing, whether it's a deletion or whether it's an insertion. What kind of things can cause mutations? Both radiation and chemicals are kind of the two big things that we think of. There's two different kinds of radiation, ionizing and non-ionizing. So what's the difference between those things? Remember, visible light is somewhere between 400 and 700 nanometers. As we get shorter wavelengths, we get increasing energy. So ultraviolet radiation and then ionizing radiation, that's our X-rays and our gamma rays. Whether you consider it ionizing or non-ionizing, it depends what kind of mutation it causes in the cell. So absolute wavelength is, kind of like, around this point. So somewhere between 10 and 300 or 400 is considered non-ionizing. That's our UV range. And then the ionizing shorter than that. So what's the difference as the kind of damage that we see? Once again, ionizing is going to be X-rays and our gamma rays. They are going to make ions. In other words, it's hard to talk and write at the same time. It's going to make ions. That means that ionizing radiation is grabbing electrons off of other molecules. So that's going to cause a ton of damage in the cell. It can damage proteins and other structures as well. But we're interested here in the kind of damage that it does to DNA. So it can do a lot of things. It can modify the actual bases. It can break one or even both of the strands. Right, so that's the great thing about DNA. If one strand is broken, you still have a template, and maybe you can salvage that information. But if both strands are broken, it's pretty difficult to salvage that sequence. It can break the hydrogen bonds. Okay, so all those are effects of ionizing radiation. Again, the most severe is that it can actually break both strands of the DNA. Non-ionizing radiation causes a particular kind of mutation that we call pyrimidine dimers. So most often thymine dimers, sometimes cytosines. It seems to happen more often with the thymines and the cytosines than the purines, just because of the structure there. But when we get enough UV light hitting the DNA, it causes two thymines, for instance, that are right next to each other in a strand of DNA to bind to each other. Remember, they're not ever supposed to be. They're supposed to be hydrogen bonded to their complementary base on the other strand. So we don't want this happening. You can see if this strand was replicated, because there's a bulge in it now, it's probably not going to be replicated properly by DNA polymerase 3. It's probably going to get maybe an insertion or a deletion in there. If that strand was being transcribed, the RNA polymerase probably wouldn't read it correctly. It would probably make mistakes. Okay, so given enough UV exposure to a cell, enough of these mutations are going to build up and eventually cause death of the cell. Okay, and you'll be doing a lab in just a couple weeks where we're going to expose our bacteria to UV light and see which ones survive and which ones don't. There are also a lot of different chemical mutagens, and I want to talk about a particular kind of chemical mutagen because I'm going to actually bring this up a little bit later in the course. But nucleoside analogs are a kind of chemical mutagen. So remember what a nucleoside is. That's when you have the deoxyribose and the base. Okay, we would call it a nucleotide if it included the phosphate as well. So that's, again, the sugar and the base. That's a nucleoside. And anything that's an analog, that's something that resembles it greatly in structure, but it's not quite the same thing. Okay, so these are things that mimic nucleosides. And what happens is that they get incorporated into the DNA, but then they're not going to be replicated properly or replicated at all, or maybe they will just cause the wrong base to bind to them instead of the correct complementary base. So causes a lot of mutations. A few examples, a couple that you may have heard of, AZT, azithromycin. That was the very first drug that had that was effective against HIV. Usually viral polymerases are really not as particular about what it throws into that polymerization reaction of replication. So AZT can easily become incorporated. This is a structure over here. You can see this is actually thymine, the base, but then the sugar here, it has an azide group instead of that 3' hydroxyl group, what we see in deoxyribose. So when that gets incorporated into the viral DNA, there's no 3' hydroxyl group for it to build on, so it causes chain termination. Okay, so it's supposed to keep the virus from replicating. And actually, there's a new name for that, zidovudine. I'm not sure why there was a name change. I think AZT is still kind of more easily recognized. But another example, 5' bromo uracil. This is used as an experimental mutagen, I believe. But you can see the structure here. This is the structure of thymine on the left. And on the right is 5' bromo uracil. It only has a difference. It has a bromine group there instead of a methyl group. Okay, so essentially, when that 5' bromo uracil is incorporated into a cell instead of thymine, it base pairs with guanine instead of adenine. So we're just going to get multiple mutations built up. And here's another example, a drug that was just approved by the FDA, I think like end of December, early January, molnupiravir. This is a cytidine nucleoside. Okay, so again, a nucleoside analog. We're trying to trick SARS-CoV-2, that virus, into incorporating this bogus nucleoside. And so what it does is actually causes that virus to make many more mutations. Those viral, again, polymerases aren't very discriminate. They will incorporate, they're not as picky as what they incorporate. So here's what cytidine nucleotide should look like. And then this is the structure of molnupiravir. So it's supposed to cut down, and this really was made headlines because it was the first COVID treatment that we could give as a pill instead of having some kind of infusion. So it's supposed to cut down on hospitalizations quite a bit, and it was approved by the FDA just by like 9 to 8 vote with the committee. A lot of people argued that we shouldn't be using this because number one, you can't give it to pregnant women because this is going to be incorporating into any kind of cells that are growing rapidly. So like a fetus, and it would introduce a lot of mutations. So obviously we don't want that. And also we have to think about, is this really what we want? Do we really want this virus to mutate faster? You know, it's already doing, we're already getting lots of different variants. So maybe not, this is what we want. The good news is about the same time, another drug came out that I think is going to be a lot safer and works really well as well. So we're going to talk about that a few weeks from now. So stay tuned. I just want to briefly mention repair mechanisms and just that in general, most cells are going to, you know, come in contact with some mutagen at some point, or, you know, their enzymes are going to make mistakes. So they have ways to fix errors. All cells do. And I think I'm not really going to go into most of these. Some of the enzymes will just kind of fix a base if it's the wrong base. Some of them will, you know, cut out portions of the DNA and, you know, make it again. One particular mechanism that I do want to mention is called mismatch repair. Because I mentioned earlier that often after DNA gets replicated, it often gets methylated as well. So here's one reason why that happens. So this is not in your book. I got this from another source. First of all, little review, which strand up here at the top, which one is the leading strand and which one is the lagging strand? Okay, you can tell by the directionality. The bottom one here, this is 3', so this actually is the leading strand. Remember, the new strand's always being added on that 3' end. And then this one must be, you know, working in the Okazaki fragments away from the replication fork. Okay, so don't always assume that we have the leading strand on the top and the lagging strand on the bottom. Okay. So after replication, here we've got the new strand is in red. And look right here, the enzyme has made an error. It's matched a thymine with the guanine. Okay, so with this repair mechanism, there are enzymes that move down that new piece of DNA and look for errors. When it finds that error, though, how's it going to know which strand to fix? Does it fix the G or does it fix the T? It's going to fix the one that's newest. And it knows which one is the newest because the old strand is methylated. Okay, it's got that CH3 group. The new strand hasn't been methylated yet. Okay, so bacteria usually methylate adenines. Okay, it knows, okay, the G, that's the old one, so I'm going to fix the T. So here, down here, it's fixed the new strand. And then eventually that new strand will get methylated as well. And once it does, then this repair mechanism is not going to work anymore. Okay. So we say, when we say it's hemimethylated, that means one strand is methylated and then the other one isn't. Okay, so the old strand is and the new strand isn't. Okay. And interestingly enough, this error-prone repair, so most of these enzymes that will fix mistakes, they do so, they need a template to kind of work from, but this error-prone repair, in case both strands get severed, there are DNA polymerases like DNA polymerase 4 and DNA polymerase 5 will just kind of make up a stretch of nucleotides without any template. So just kind of throw them in there. It's kind of like the half-court shot right before the buzzer. Like, it probably isn't going to go in, but maybe, you know, 1 out of 1,000 times it will. That's kind of what SOS prone repair is. It makes a lot of errors, but maybe somehow that offspring could possibly survive. Okay, stop me if you have questions. All right, we're making good progress here. Just one more thing we need to talk about. So there are other ways that the DNA of a cell can get altered without mutation, and that's by some kind of horizontal gene transfer. Right, so y'all know what vertical gene transfer is. That's when a parent cell splits into two and gives its genes to its offspring. Horizontal means it's in the same generation. Okay, so one cell will be able to pass some of its genome on to another cell. So there's three kinds of horizontal gene transfer: transformation, transduction, and conjugation. Okay, so we're going to look at them one at a time. So transformation involves cells taking up DNA from their environment. And usually you think, well, when would there ever be DNA in their environment? If they are around other cells that have died, when a bacteria cell dies, their cell wall starts to break up and contents from within the cell can leave the cell. So it's possible. We don't think this happens very often. In general, we think horizontal gene transfer is not very common. Maybe only about 1% of cells might get some DNA this way. But it's possible, especially for certain species, to be able to take up DNA from cells that have died in the environment. And we actually can make cells able to do this in the lab. So we can treat them with calcium chloride, or we can run a current through them to make them more likely to take up DNA that we want them to, see what happens when they take it up. Okay, so this is kind of the basics. There was a huge article in the Wall Street Journal last weekend about using RNA for different therapies. Of course, we know it's promise. We've already seen that for vaccines. But there's a lot of potential for making lots of different vaccines better by making RNA vaccines, like our flu vaccine. It could be better if it was an RNA because we could make it up really quickly rather than it taking like 6 months to do it. So we could match it up with the strains that are in circulation right now. So if we had a flu vaccine that was, 80 or 90% effective, more people would probably take it. There's also potential to use RNA to treat cancer or to treat lots of other diseases. There's a lot of hurdles with getting cells to take up DNA or RNA. So that's, we're seeing it, just a ton of research really frantically going into that right now. So let's talk about the experiments as we end here that gave evidence that transformation actually happens. And this was done by Frederick Griffith. So everyone saw, we like to look at kind of really important experiments. Frederick Griffith did this in 1928, which is astonishing because we didn't know very much about DNA back then. We didn't know its structure. So to think that, he even understood what was going on here is pretty exciting. He took two different strains of strep pneumoniae. So this is a species that causes a really severe pneumonia. In humans, it's deadly like 50% of the time without treatment. So it's a really nasty pneumonia. And he had two different strains. He had the wild type strain, which he called strain S. So it was shiny. The colonies made a really nice, vigorous capsule. That's what they normally do. He also had a mutated strain, which he called strain R for rough colonies. And these was a strain that couldn't make a capsule. So when he inoculated the live cells of strep pneumoniae into a mouse, the mouse usually died, okay, got pneumonia. If he heat treated some of that strain S, so in other words, he inoculated the mice with dead cells, the mouse lived. That makes sense. If he inoculated the mutated strain that didn't make a capsule into mice, they usually survived. So that capsule was necessary for that organism to establish itself in the mouse host. So then the really weird thing, and just give me a minute here, is that he took the living strain R and mixed it with the dead strain S. And oddly enough, that killed the mice. And he was able to harvest the living strain S. Okay, so that gave evidence that that the living strain R was able to take up DNA from those heat-treated dead cells. Okay, so that's the evidence that we have that transformation takes place. We found that he could also do that in a test tube and have it work really well. It didn't have to happen just in the mice. Okay, so we will finish up cortisol gene transfer next time. Start working on that homework if you haven't done so. We will move on to viruses on Wednesday as well. Okay, have a good day.